All right. Good afternoon, Brodies. Great to see everyone here. The room is packed, and for those of you that are joining from the overflow rooms, welcome. And those of you joining online as Brodies or guests, really great to have everyone here. This is an exciting moment to reflect on the first 20 years of the Broad. I want to also give a special, I don't know if I see here in the room quite yet, but Garen Riley, who's president of the Broad Foundation, is here with us today. The Broad Institute, of course, wouldn't have existed without the visionary philanthropy of Eli and Edie Broad and the Broad Foundation, so it's wonderful to have Garen here with us today. You know, part of our culture at the Broad has always been about always being forward-leaning and thinking about what's the next great thing that we should be working about and not just talking about what we've done in the past. But once in a while, it's actually a really good idea to do that. And if you think about what has happened over the past 20 years at the Broad and in our whole field, it's been pretty remarkable. So I think, you know, we deserve the opportunity to do this and just to think about how far we've come and how unimaginable really so much of this was 20 years ago. We've got an amazing panel to reflect on this today, known, I'm sure, to everyone here, but for those of you, particularly those that are joining from the outside, first and foremost is Eric Lander, our founding director of the Broad, whose inspiration and vision made the Broad possible to begin with and led the Institute for the first 17 years. And I think Eric, like no one else, is going to have perspective on the past 20 years. I doubt any of us had Eric's sense of vision of what could happen as a result of the Human Genome Project. So great to hear from Eric. We have Stacey Gabriel, who is the Broad's EVP of Platforms and Scientific Execution. I think Stacey has been a living example of being a scientific giant, not just as a faculty member, but as a staff scientist. And this is, I think, a special thing about the Broad community, having so many really talented staff scientists in our community, and Stacey has been exemplary in that way. David Altshuler, who's one of the core founding members of the Broad, started the Medical and Population Genetics program, established really this very deep connection that the Broad has with Mass General Hospital, and David really helped establish some of the foundational culture of the Broad that really continues today. So welcome back, David. David, for the past 8 years or so, has been Chief Scientific Officer at Vertex. In the front row, we've got a bunch of longtime Brodies. First and second rows, a couple longtime Brodies who I'm hoping will jump in with your perspective as well as we go along. But really, all Brodies who are here and have come through the Broad over the past 20 years are really part of this celebration. I got the number recently that 23,000 people have passed through the Broad over the past 20 years. And so when you think about that number, it's really pretty stunning, and it's a lot to be proud of. Obviously, the vast majority of those people out there in the world, but I'm sure they took a part of the Broad with them. If you think about the resources that we create, the data that we create, that's another way that we've had impact in the community. We're going to talk a lot about the sequencing of the first human genome, but the rough calculation is that the amount of sequencing information that we've generated is 3 million times more than that initial sequence of the human genome. I think what really a shared theme across all that's happened across the Broad for the past 20 years has been this idea of bold ideas, reaching higher than is comfortable, and doing that with a shared sense of vision and a shared sense of community purpose. And there's like, what could be more satisfying in life than doing that? And I think having that shared vision and purpose and thinking boldly, I hope that continues, not just for the next 20 years, but forever. So the discussion today is going to be moderated by Lee McGuire, our Chief Communications Officer, who everyone knows and loves.
Keep us together.
But to get us started, we've got a short film to kick things off. So let's roll the tape.
Both Edie and I are most pleased to be here to announce the creation of the Broad Institute. The purpose of the Broad Institute is to create a new type of biomed research institute to build on what has been accomplished over 13 years of the Human Genome Project. Let me tell you how we got here.
Human genetics was virtually impossible in the 1980s. Elaborate, complicated molecular biology, incredibly slow sequencing. I was a young scientist, drawn into the Human Genome Project by the dream that all these things that took armies to do could be done by single graduate students. And it happened.
We are here to celebrate the completion of the first survey of the entire human genome.
I want to be sure you realize what a remarkable thing we pulled off. I hope you also know that this is history. Whatever else you do in your lives, you're part of history.
Once a warehouse for beer and popcorn sold at Fenway Park, now transformed into an international scientific landmark as the leading sequencing center in the world.
It's an amazing Boston story. Beer and popcorn warehouse becomes Human Genome Project.
The first time I walked into 320 Charles was actually my first day on the job here. And I was just young and naive, and I wore high heels, and I don't know what I was thinking. It took me 10 seconds to realize I had made a grave mistake.
It was built as a bottling plant for Budweiser. It was built with sort of industrial scale in mind, and the neat thing is that sort of what this was built for kind of continued in a way, which is industrial scale processing of something, right?
Through the work of so many people who have that kind of expertise to bring those sort of manufacturing principles into science. And it just felt like this is like nowhere else.
There were few walls. It sent a real signal for how people would interact in open spaces. And you see this now in modern research buildings everywhere. And 320 Charles was really the place where that started.
A lot of people thought that once the Human Genome Project was done, we could go back to business as usual. Anything but that. And we realized that it was not a one-off, it was a new way of doing science that could propel individual creativity and individual discovery by removing the barriers to smart young people taking on hard problems.
The sequence of the human genome was a fine start, but it's only a start. The goal of the Broad Institute is to empower the next generation of biomedical scientists by providing a setting in which they can come together to tackle those larger collaborative projects that can't readily be done in the traditional setting.
Early on, there was a feeling that it wasn't going to be that traditional model. There was a value placed on building things for the common good.
There was a certain energy in the air. We were really deeply committed to saying we should give the data away immediately in its entirety. Not just the bits that we publish on. That's now the standard that we all take for granted, but it didn't used to be that way.
We were going to take an approach that nobody had taken before, and without working collaboratively on some of these big projects, we wouldn't be charting the path forward that we have been.
We approached everything as if anything was possible. There was nothing going to get in our way.
And to have that many people all together saying, we just must make all of this succeed, and that much optimism and energy, it was exhilarating.
I remember thinking, this is the future. This is what we're going to use in medicine. We have that sort of blueprint, we have the roadmap, right? And now we just have to go and learn from it and apply it. Couldn't have been more important, I think, to have the human genome.
A toast to this remarkable group, to the work we've done, to the work ahead. Hear, hear.
Hear, hear.
Okay, so to get us started, let me take us back then to 1999, which is that speech in the Atlantic Room at 320 Charles. There's a lot of 320 Charles people here in the room today. I could tell in the chuckles in the video when we saw kind of what we all looked like in 1999. Just basically the same for most of us. Sheila Dodge, you saw her in the video, she said that was the moment where she decided what her career was going to look like. And she's still here, right? She's in the front row right now. We have a lot of Brodies in the front couple rows who've been here a long time. And as we go, by the way, everybody there, put your hand up and contribute to this conversation as we go. You're welcome to do that. But Eric, I looked at the transcript of what you said in that room back in 1999, the Atlantic Room, 320 Charles, and you said, it's not like going to the moon where we visit occasionally. This is something that every student, every doctor uses every day in the next century and the century after that. It's something to tell your kids about, something to tell your grandparents about, grandchildren about. I got my transcript wrong.
Grandparents too.
Your grandparents would also be interested. Your grandchildren will be interested too. It's something to be tremendously proud of. So take me back to that moment, 1999, 320 Charles, former beer and popcorn factory, and you're changing the world.
We had just finished 1 billion bases as a whole Human Genome Project. And it was a little exhausting. We were working incredibly hard to get there, and it was important to stop and say, this was a landmark, it was a big deal. And we all met in the Atlantic Room over at 320 Charles Street to celebrate as part of this international network that was celebrating the billion base bash in many different places. And I must say, I wanted to give the sense of that sweep of what this was going to be part of. I was so far off in my imagination of what was possible. I was trying to sort of say, look ahead, this is going to be historic and change the world. And I was off by at least 3 or 4 orders of magnitude in what could become possible. And I think the important point about talking about history is not history, it's that it's the same thing today. That all of us fail to imagine just how quickly progress is going to go. I was 42 at the time. I'd started the Genome Project in my early 30s being involved. And what you see over the sweep of the next 3-4 decades in your career is just amazing. So, for example, the billion base bash, in the last 3 seconds, the Broad has completed 1 billion bases. That's just like mind-boggling. And everything that flowed out of that Human Genome Project that we'll talk about, I hope people in the front row raise their hands and jump in, but unimaginable. There was a debate about, earlier, should we sequence the human or the mouse? Because we clearly would never be able to afford to do both. But of course, we did the human and the mouse and the chimp and the dog and the rat and 200 mammals and all that, and cancer genome sequencing, collecting all the variation in the human genome and using it to create genetic mapping and GWAS applied to zillions of diseases, and onward and onward. Single cell biology and genome editing. And I was pretty enthusiastic and optimistic, but I could not have imagined how much it would compound and compound. And this new model that we were all building together of taking on things that you couldn't do in a traditional setting, but you could do working together, it was beyond what I ever could have imagined. And I hope everybody here, whatever age you're at, realizes you're going to feel that way about the future when you get there.
Can I make one comment just to give context? I was a MD, PhD, a doctor at Mass General, and I had arranged to work with Eric. You'd accepted me in your lab. It was 1996, and you published a paper in Nature Genetic Science called Global Views of Biology. And I read it, and Eric said in this paper, this guy I was going to go meet with, it had like 10 things that would happen. Oh, good, you got it right here. So it was like, honestly, but the main point, actually I want to check if they all happened, but anyway, like every one of them. The amazing thing is that when I read it, in all seriousness, I thought, I don't know if I should go work for this guy because he's crazy. That's actually what I thought. Because
He's told me, yeah.
And so, but the amazing thing, so that's 28 years ago. Today, it had, actually it wasn't really a trade, it was random, but I'm here and Fei Chen gave a talk at Vertex, I'd invited him months ago, and I was listening to him talk about single cell sequencing of millions of cells, and I thought, in 1996, I don't think even Eric proposed that there would be like someone starting their lab saying, well, after we sequenced 8 million cells, then we decided, and so this is documentation, A, that it seemed crazy at the time, and B, that like actually more has happened. I bet it says like in the next century or something like that. You know, it's crazy. Everything in that, I think, happened.
Everything did happen.
It's amazing.
One thing you said in that paper, Eric, was we might have to sequence more than one human genome.
Oh yeah.
To really figure this all out. It might be necessary to do a couple.
I think I said more than a couple. Yeah, no, we were into, but what we had to do, and this was with David, is we had to come up with cockamamie ways to get lots of human genetic variation when we could barely afford to sequence. We came up with reduced representation sequencing and other tricks, which there's no point in reading anymore, but it actually gave us the first big collections of human genetic variation, because every bit of the way you always feel like we're so constrained. How do we live on this edge? How do we advance it? And you just keep doing that, and suddenly things fall together. And it's no different today. That's the biggest take-home you should get, is it's no different today.
Can I just add a quick perspective, Eric, on your feeling like you were off by 3 orders of magnitude, in one direction. At the time when you said that, I think most people thought, kind of as David alluded to, that you were off 3 orders of magnitude in the other direction.
Right.
Because at that time, the Human Genome Project and the early stages of genomics were in this obscure corner of kind of a wasteland of Kendall Square. And so the idea that, in the future, every biologist is going to be slinging genomes around and working with this kind of genomic information on a daily basis, that was so far off from what was happening. It was just hard to imagine.
It's actually a good segue to a video message we received. So one person who really wanted to be here couldn't be here, and that is Francis Collins, former head of the NIH and the NHGRI, who's one of the many individuals who really pushed this forward back in the mid to late 90s. You've all worked with him very closely. He's been here and played his guitar in this very auditorium before. Francis sent us a video message taped by his wife in his living room, and let's watch that now.
Hello, and congratulations to the Broad Institute on this 20th anniversary. Today, I hear we're telling origin stories, and I'm sorry I can't be there in person, but I have some stories. It was 1990. It was time to start building a genetic map of the human genome. The Genome Project had just gotten started, and RFA was put out to build microsatellites. Probably some of you have no idea what that is, across the whole genome, but actually parsing it out individual chromosomes at a time. The review group was chaired by, guess who, Eric Lander. Eric arrived, looked at all these proposals, and basically concluded the whole premise was flawed. Why on earth would you want to do this one chromosome at a time when you could just do the whole genome? He basically recruited other members of the review group, me included, to agree with him. The whole thing kind of blew up. I thought maybe Elka Jordan was going to have a stroke, but somehow we took what had been proposed, reshaped it. Originally, the investigators hadn't planned to quite do it that way, but it worked. A classic moment in the Lander scheme of how to make the Genome Project really about the genome and not smaller parts. And then we had to build a physical map. The first physical map of the human genome, done by the French, Daniel Cohen, was pretty hard to work with. We had to have something that was more public, readily able for people to use. So what happens? The Whitehead MIT Lander team builds the Genomatron, an amazing automated system that was going to build a map of yeast artificial chromosomes based on STS mapping. Look that one up if you're not sure what it is. And sure enough, you had to do PCRs in numbers so huge, a waffle iron was built to do that. Okay, but then it was time to move on to sequencing. And well, we needed to build a factory. And there in 320 Charles Street, a rather undistinguished warehouse, putting together in about an amazing 30,000 or 50,000 square feet, a factory with a factory mindset. I went and visited it. I found the email from this in October of 1998, and I was impressed. The focus on speed, quality, and cost, the big three, was already very much the mindset. And I noted then that the folks at the Genome Center, Whitehead MIT, led by Lander, were planning to submit a new proposal, and it was going to be, quote, vigorous and ambitious. Well, to Eric, throughout the Human Genome Project, you have defined vigorous and ambitious. You always push beyond the limits of what any of us thought would be possible. You infected all those people around you with that same outrageous attitude. They've been landerized, which I think is an irreversible condition. When historians write a few hundred years from now about the Human Genome Project, I think they will call it the most ambitious and most successful life science effort of its kind at the time that it was conducted. And I think they will also recognize it could never have succeeded without this creative, collaborative, scrappy group in Cambridge, Massachusetts. So congratulations to all of you.
Nice. I miss that guy.
One of the people who helped create that scrappy culture at certainly 320 Charles is Stacey. Stacey, I know you came on board a little after the others did, but tell me about your first impressions of this place and what you walked into and what it feels like today.
Yeah, so the 1999 speech was very soon after, maybe just a couple months that I had, I'd come here. I had just left my graduate program in Cleveland, Ohio, which was very much a traditional, do things sort of the old way, a classic sort of academic setup. And I came here because I saw that first Affymetric SNP array paper. And I thought, well, I met Eric in graduate school. And then I saw that paper and thought, wow, if he's doing that, if he has that SNP array working, and I could do that kind of mapping that took me 4 years toiling away with that map that Francis talked about, if I could just do that, I could have completed my whole PhD in a week's time, basically. And so that clearly seemed like the right thing to do because I never wanted to do that PhD again over those 4 years of microsatellite mapping. And so, yeah, arrived here and saw it. And I was like, oh, so yes, this is how they're doing it because they have this infrastructure, this building, this way of thinking and not being afraid of doing anything. And so, yeah, I knew it was right away and worked closely with David and Todd and learned my way through it. But it was amazing.
So you should say something, though, about your interest in being a professional scientist. Because you came here differently than almost anybody would.
I came here against the wishes of my PhD advisor. So those of you in graduate school, you can imagine how that felt for someone who said, don't go do that. Let me hook you up with a nice postdoc position in the lab at MIT if you want to move to Boston, although I think that's a terrible idea too. But you should go do a postdoc. You want to take this approach. You're still training. And I said, well, I want to go. Eric said I can come up and talk to the team. And so I sat with Eric in his office, and I made a big speech about how I didn't want to do a postdoc, and I didn't want to do an academic path. And Eric said, you want a job? And I said, yeah. And he said, well, all right, come on board. And then it's I would say history from there. I've always found something to do here. And it's been great.
So in a second, we're going to show another video, which I think is a great video that shows kind of the early days of the Broad and the spirit of this place. But as we get into that, so the Human Genome Project is more or less completed. You all know there's a ton of work to do to understand now what it is you have and apply it. And the idea of the Broad Institute sort of begins to emerge from that. Tell me about that moment in time for you, but also the culture you thought would be needed to succeed at a place like this in like the early 2000s, late 90s era. Give me a little bit of that.
Well, I'm going to turn to David, actually, because the Broad Institute was largely conceived in one physical location, my back porch, over the course of a summer. And we had dinner, Todd, David.
It was a Tuesday. It was always Tuesday.
It was always Tuesdays, right? And Connie made dinner.
Yeah, it was always good.
Yeah. And so it was a small number of people sitting on the back porch. And the things we talked about how do you create a scientific future and a culture and an institution, I'm more interested to hear David's, because you have this, you're very thoughtful about these things. So I'm tossing to David.
So first of all, I think all of us had the experience that when we found each other, it was like, oh my God, this is it. But, just one quick story, because I think that maybe people don't always realize the risk involved. I had done a postdoc with Eric. I had finished it. I'd been offered jobs at different universities, and I wanted to be and was a professor. And everyone said, you cannot keep working with Eric, because like he's your postdoc advisor and you can't work together. And I had offered great jobs, lots of startup, and I was like, there's no way I can do what I want alone, but also I kind of want to keep working with all these people. We're having fun. So the reason it was so fun was we had a shared mission, which was, in the case of the Genome Project you described, but I was really interested in human genetics, as you were, as Mark Daly was, Joel Hirschhorn, John Ryu. There were a set of us who were there at the beginning. And we worked together because we had a shared vision. We worked together because we had shared technology, both the technology and the statistics. And we had created this group that was called Medical and Population Genetics back in '99, I think, that was meeting every week. And it was not like the other human genetics labs, which all like would hide from each other and steal from each other. We were all like, if you wanted to come to MPG, which I chaired from '99 till 2014, what I'd say at the meetings was, if you come to this meeting, you have to share and you can't steal. If you ever take anything from this meeting and do it, you are not welcome anymore. And the amazing thing was it never happened. Never happened in 15 years. So we were sitting on your back porch saying, how can we build a place with that culture? Now, it wouldn't be one thing. It might be cancer, because Todd was doing something very similar. It might be infectious disease. I see Pardis over there. It might be, any one of, we had the genetics of different species to understand evolution. And I could go on. And I think to me, that was the idea of the culture, was the most important thing was that we'd have groups of people who were working together, faculty. We always knew it was the students and postdocs who actually did the collaboration. And then staff scientists who were like partners, and we'd sit in these program meetings and we'd come up with ideas, and then you build the platform. And it was a virtuous cycle. And so we sat there and said, how do you do that? And the last things I'll say, I talk too much, but the last things I'll say are, you had to know each other and trust each other before you collaborated. And I think that actually was the secret of the Broad. That and free food was that...
Don't underestimate the free
No, I'm serious.
food.
That what we talked about on your porch was, why did we all collaborate together so well? Because we'd all worked together in the same building and we worked on a shared project. So, I could tell a story about Jay Bradner and Steve McCarroll. You remember sitting outside your office coming up with a project, just because they were sitting next to each other. That was the culture of the Broad, and I hope and believe it still is. And the idea was, if you want people to collaborate, you can't wait until you need them and then call them up and negotiate a contract. What you have to do is build a community where everybody trusts each other and understands, the shared interest, so that when it happens, you can move fast, you can move effectively. And so everything was about, like, how are we going to find that community? And I remember we talked about a semi-permeable membrane, which was that we'd be connected, amazing thing that Eric was able to arrange and negotiate, Harvard, MIT, and the Harvard hospitals. And we realized that it was open to everyone, but if you didn't like the culture, if you didn't want to be a team player, maybe the semi-permeable membrane, there might be, I don't know. You might just diffuse out. Right. But if you liked it, you could work together. And that was the secret, was to create trust and shared vision of the program, the faculty, the platform, and then the virtuous cycle that led to thousands of papers and discoveries from each of those, from those groups.
Can I add to that also? There was an early culture of don't ask permission, ask for forgiveness.
Yes.
That was really important in the pre-Broad days. And so I remember the discussions on Eric's back porch around saying, okay, that's been really good. If there's something that people want to do, they should just be able to go do it and not have like months of committee meetings to talk about it. So how do you create an institution that would become the Broad Institute that does that day in and day out without killing that kind of entrepreneurial spirit in the process of institutionalizing it? And so that was super critical in sort of maintaining that scrappy kind of culture from the very beginning.
If I can add something to Todd's point, the traditional way of doing this is high-level interinstitutional, the heads of institutions doing it.
Death. Don't do that.
The Broad was organized at the ground level, at the grassroots of people interacting. And what we ended up doing was negotiating an agreement amongst all the institutions that created just a free trade zone where people could do that interaction. And they didn't have to go up to the leaders, ask permission, and do that, because there was one kind of master way of all these institutions working together. But I forget how important that was, because we got away before the Broad Institute because nobody was paying attention to us. So there are all these people working from different institutions together, but nobody paid attention. We had to somehow come out in some way that would let it keep going.
I think Lee probably wants to move on, but I'm going to say one more thing, which for all of you, just to understand the opportunity that's here for you. The way the program, and I'll use MPG as an example, Stacey ran the platforms, and Mark Daly was sort of the leading statistician, and I was whatever I was. And we would, of course, there were lots of other people, who are still here. But what we realized was, first sort of organically, and the same thing was true in all the other programs, that if you had a platform, you could collect preliminary data. If you collected preliminary data and spent a lot of time debating amongst yourself what the best idea was and put the best team together, you could get a grant or you could raise money. If you raised the money, you could then not only do that, but you could generate more data to do the next one. And it was that you were so much more effective that I remember, I could give you so many examples where people were like, which is really sort of sad, they would, in the rest of the human genetics community, they'd be like, you guys have to stop because it's not, I'm not kidding, it's unfair. And we were like, what are you talking about? And it was like, you're moving so fast, how are the rest of us supposed to keep up? And my answer was always like, we're doing this for sick people. No, I'm serious, we were studying the genetics of disease. You want us to slow down to find the genes for a disease that will then take 20 or 30 years to figure out how to cure it so you can go as slow as you want. And people, no, I'm serious. But for all of you.
He made a lot of friends by doing that.
No, I did actually. I actually did.
I know.
Because what happened was we invited them to join us.
Right.
So what happened was they said that, and we said, well, why don't we just collaborate? And then we all started getting more grants, and the whole field accelerated. But the reason I wanted to mention it was not to talk about the past. It's like I'm on the board of scientific counselors now, and actually this is my 4th out of 5 days being at the Broad. I'm on the FOSSI committee. It's fantastic. And once a Brody, always a Brody. And but what I was going to say was I listened to all the talks, and I was like, oh my God. There's so much incredible stuff going on here and so many opportunities, and people would be talking about it, and you're just like, I hope everyone sees that you're in a place where you're inventing things. Some of you have biological problems or medical problems you're more focused on. Some of you are inventing technology. Some of you scale things for a living. Some of you do analysis. If you all work alone, you're like everyone else, and you're not going to do any better. But you figure out how to work together, you are going to not only accelerate your own work, you're going to actually accelerate the world. And that, if you don't understand that, you're going to miss out on the opportunity and go slower. So people sometimes think if we collaborate, we go slower. I mean, if we collaborate, we might have to split the pie. You can make the pie, 3 million times bigger?
Yeah.
So I just want to make sure everyone here understands it, because having just come from the BSc, you are just in this incredible environment. Just make sure you take advantage of it the way I described, by working together to solve important problems.
So that actually tees up our second video really well, which is early on when the Broad now has launched, and here we are, and we have this amazing opportunity in this community. The decision was to build a place that can think big and move quickly in ways that had never been done before. So let me show you what that felt like 15 years ago.
5 years ago, Eli and Edie Broad came to Boston to announce their support of an experiment. 4 years ago, we launched that experiment.
From day one, it was always, what's the next task? Who can help do it? How do we solve the next problem?
So we'll have to run some sort of a conduit.
That's always been the culture.
The team approach, that you have individuals who are experts in their particular field and together working on a large project that no one individual could undertake.
We underestimated how hard it would be to understand the function of the human genome, and we also underestimated how important an understanding of the genome was going to be.
There was a concept put forward that we should build something called a haplotype map. So across the whole genome, across different populations. To develop the map, we're going to have to perform a few million of these genotyping reactions. Those numbers at the time were extremely daunting. If it wasn't overwhelming, it wouldn't be the Broad.
We were pushing things so hard. We were on what we always called the bleeding edge of the technology. We were the first ones to get a new technology and say, it's not working, go back, fix it, try this.
And these were kind of like the early days of robotics and automation. Our engineering groups were building their own custom robots that work as much as they could in their early infancy.
We had a giant machine shop downstairs with lathes and mills and metal filings flying everywhere. We really did have all the things in place to sort of meet those sort of challenges as they came. But there were times when, we had to do something but it was going to be kind of clumsy.
There were weeks where we had cords running through the building because we were having power outages in Cambridge pretty frequently. We had to literally connect it to kind of a makeshift generator. And so you'd have to step over the cords as you were doing your work.
Grants were done on paper and taken to the FedEx. We had this big grant, which became the Microbial Sequencing Center eventually. We got like 4 feet of snow and FedEx wouldn't deliver, so we bought 2 train tickets and 2 people actually carried them down to Bethesda to submit them.
We were part of large consortia projects. One such project was the Human Microbiome Project. At some point in the project, all of the other genome centers had started producing data, and the Broad was kind of called out for lagging behind because we really believed that it was important to automate the process so that we could do it reproducibly at scale for a long time and at high quality. At the end of the day, we submitted all of our data before anybody else finished, and all the subsequent studies showed that our data was the highest quality of all the centers that generated data.
A lot of those projects, they've all been helping ask the next question, and it would be the justification for the next effort.
We create tension, right? The right amount of tension within the organization, and we're constantly kind of pushing the whole organization because this is like the culture and this is how we think about it, and we're fearless about the things we can take on.
We've always had this sort of mantra of we don't know what we don't know yet. And that's something we have to continue to work through.
This idea of people being driven by wanting to make discoveries that will really impact the world, taking on something that's bigger than themselves, that's the Broad spirit as far as I'm concerned. That's the spirit and the culture and the energy that I think will sustain us into the future.
So Eric, I saw you looking at some of the front row people.
Yeah, we should get them involved.
Yeah, not just the early projects, maybe some more recent ones. There's some folks in the room who have been involved in some of those big, ambitious ideas that no one else had tried before that we thought, worth a shot. Pick someone, and maybe they can they can tell us about it.
Well, Brad, I remember as you were an assistant professor, and you were applying for the epigenome roadmap, something or another, and you came into my office and you said, you had this whole vision of what needed to be done with epigenomics, and you said, the problem is, I'm just a starting assistant professor, and all the other people applying are 800-pound gorillas, and I'm not going to get this grant. You can take the story from there.
I know that's your favorite part of the story, but I think the story's even better than that because, my story, origin story, is very much parallels the Broad story, but it starts, I think, in 2000 when I was obsessed with chromatin, and I went to work with Stuart Schreiber, your fourth compatriot founder, who had, we didn't understand chromatin, but he had just isolated, cloned the first chromatin repressor. And we had to figure out where this thing was going and what it was doing. We didn't understand chromatin at all. So, I went to the Center for Genomics Research. We had some microarrays, and we could just barely do this in yeast. We made microarrays with yeast promoters, and I did all my analysis in Excel. And then you guys released Draft 21 and Draft 22. And there were these microarrays. And I knew that chromatin is much more interesting in metazoan organisms because it opens and closes genes and it specifies lineage. And I was working in yeast and missing out on this. So, if people watch the Celtics, there's the coach says timing is everything. And I said, Stuart, this is what we should be doing. I got a problem. There's no way we can do the analysis. And he said, Brad, I happen to be starting an institute with Eric right now. And would you like an introduction? So I got a 7 a.m. meeting. We went to breakfast. I doubt you remember, but we went to breakfast. And the rest is kind of history. You weren't working in Excel. You knew how to handle entire chromosomes. You invested big time. I moved my lab over to the Budweiser factory, actually, remember? And for a little while, and then I started my faculty job. We built the ENCODE catalog. We figured out how to do chip sequencing. We found all those enhancers. And I'd like to say that I planned it all, but clearly couldn't have planned it all. But I guess the only advice from me is you got to be in the right place at the right time, and you got to see it when it's presented to you. And I think that's what the Broad was for me, and it's just been a delight for 20 years. So thanks for letting me share that.
Cool.
It's more effective if you call on them than if I call on them.
Really?
Probably.
Well, I mean, I'll do a little, but everybody should. I mean, people should just volunteer. But I can't help but turn to Sheila, who has, there is no one in the world like Sheila. I find myself describing Sheila to people, all over the world. And you came here, and what you've done through genomics, and then, of course, through the pandemic, and just your ability to just take on anything. It'd be great to get your perspective on all this.
So I have sneakers on today, not high heels, just for the record. So I actually started here as a temp while I was still finishing my undergrad, and I was doing M13 transformations in the lab for the Human Genome Project by hand. We hadn't used thermocyclers yet. I think my experience here has been amazing in the sense that we've always had a culture of fearlessness, and I love that because I like to take risks and I like to, get a lot of things done. And I think it's really been the team, right? Like we've been able to work as a team and take on these enormous challenges, no matter what sort of the barriers were in front of us. And I think, with really strong leadership and really strong mission, that has sort of been an unstoppable combination that I've, loved my whole time here. And I think, we're sort of still going, right? There's still so much to do. We talk about everything we've done, but there's a tremendous amount of work to do. And I think the fearlessness continues. So, I think when we think about the scale of the Human Genome Project, Danielle and I were talking about this, but she's done the calculation. We were actually liquid handling wise at a higher scale back then than we are now today. And so, we know how to do that. We could scale anything. And I think that's been how we've sort of thought about all the projects we've done over the years. And that's been an exciting thing to be a part of. And it's been great.
Yeah. Thank you, Sheila. Oh, go ahead.
I'm just going to add, and amongst the many things that I'm just in awe of is you're finding the bugs. There was I forget exactly what it was.
I know what story you're going to tell, but go ahead.
No,
I want to see if I got. No. I want to see if I know what story you're going to tell. Go ahead. No. I don't know. Go ahead. It's about the thing left on the porch. Remember, there was something with the sequencing data was off, and you guys traced it back through Six Sigma to there was a box left in. Was that what your story you were going to tell?
That
there was a
wonderful.
box. There was
So Sheila should tell
that is the
story.
most. Right, I still tell that story.
I
love
thought
that
you were
story.
talking about the time that there was a raccoon on top of a sequencer. That was a little different.
That's not nearly as good as the box that was left overnight.
NEN tracing things back to Ireland.
Yeah, a box overnight.
And then I also think about your
T5.
Right.
Yeah.
Sorry?
It was Templify.
Yeah, but it was also that
Oh.
first they were. If I remember correctly, we had all the matrix sequencing, and there was live analysis, which most companies don't have, where all data is being. Isn't this right? All data was being identified, and there was something weird in the data. So you could have stopped everything, but it was $1 million a day or something like that. So you probably wouldn't do it. So the question, because you'd matrixed everything, so you could analyze and figure out what was wrong based on the signal. Then you went back and figured out what reagent it was. The only thing in common was this thing. Then you worked with the company, and it turned out it was isn't this right? Do you remember this?
Yeah.
And then it turned out there was a box. Yeah.
We should let Sheila tell.
Yeah, no, I mean, the reason I'm is it this one or that one is because we do this a lot, right?
Right.
There's a million examples. The one that comes to mind that was took us a while to figure out, and I remember I had to go tell Bruce Buren every day that everything in the lab had failed again. And he would say, okay, let's try again tomorrow, and let's fix one thing at a time. But we started staring at the instruments and watching and looking at all the data, and we realized that these really expensive reagents we had on the machine, the tips were coming, there was sort of a bug in the instrument. The tips would actually go into the bleach bath first before the reagent. So it was an enzyme. So basically kill the enzyme every single time we put it into the plate. Took us a while to figure that out, and that was, kind of one example. I mean, there's many examples. I thought you were going to talk about the blueberry juice you told me to add to all the the samples at one point, because we had an artifact pop up in our exome data. Stacey will remember this 8-OXO-G artifact.
That was the one I was going for, yes.
That was, yes, so that was a problem that the samples were getting oxidized during the actual shearing step. And it took us a long time, and I think that was when I would have to go tell Stacey and Eric that nothing worked every day for a long time, or that it worked, but we were seeing this artifact, and then one day we realized it was oxidation, and Eric said, "That's great, just add blueberry juice to everything."
It's an antioxidant.
It's an antioxidant. And she said, "Okay, we'll go look that up and see if that'll work." But we did find a way to actually change the buffering, that actually reduce the oxidation, and we got everything back online.
But you've done this again and again, and the whole team has done it again and again, of just debugging problem after problem, like relentlessly. The one other story, and then I'll let Lee go on, was because you guys, normal labs don't do this. You have quality control everywhere. And there was a time that there was a problem with the quality control. I think it was the Templify or something, but whatever it was, a problem with quality control. And the Broad sent out teams to 3 different locations where stuff was manufactured to find out what the problem was. And the team that went to Ireland found that the stuff that was being taken out of the cold room, there was a standard operating procedure for how long it had to sit in the cold room, was being mixed in a cold vat that had been taken out of the cold room, but there was no SOP for how long the vat could sit on the bench. Yeah. And therefore it heated up and it caused the reaction to start. And you guys got New England Nuclear to refund us $1.5 million.
That was the story.
For debugging their process.
Yeah, we found that.
That's the story I was.
That was yes, exactly. And this is just totally routine. Anyway, carry on.
So Stacey Gabriel spent a lot of time interacting with industry over the years. Is this normal? Like, is this a normal thing that happens in an academic environment, or is this a little unusual?
Nothing here is normal. No, I don't know what you mean by normal. Yeah, but I have a good troubleshooting one. This was during COVID, and it's just an example because David, when you said we didn't press stop, like we didn't stop, like that triggered in me that actually because of the job that Sheila and Danielle and the team have done in training the team for like using real like manufacturing principles and applying those to science, they actually do press the stop button sometimes very hard if something's going wrong, and it's a hard decision to make because I mean some of you all too young to remember like the Lucille Ball thing with the candy going down the thing. And how they stopped it, but then she had to eat all the candy, something like that happened, right? So like when you're in the lab, and especially during COVID testing, when we had all those like plates and tubes flying at us, we saw a problem in the middle of the night, and one of these supervisors that were trained knew that, I gotta stop this. So press that big button, everything ground to a halt, right? And then the troubleshooting ensued quickly after a lot of panic. But I mean, that's not normal in an academic environment to have that sort of that kind of rigorous training and that, courage that you are going to, do something hard like that. Yeah.
Let me spend a minute on COVID testing, because that's a really good, a more kind of current example of how the culture that was established and the norms that were established early on in the Broad really benefited this organization and really the entire country when there was a kind of a crisis we had to help confront. COVID testing, y'all kind of know the story. Probably know the story. We ended up doing PCR testing for 37.5 million people during the pandemic. Almost every public school in Massachusetts, almost every institution of higher education on the East Coast and into the Midwest came through the Broad Institute. This is at a time when there wasn't a lot of COVID testing available. You didn't get the things in the mail from the government, and people wanted to go back to school and be back, interacting with other people after quite a long lockdown. The GP and the Broad coordinated quite quickly over a long weekend, built out an infrastructure, got the right permissions, worked with the government, figured out how to pay for it, figured out how to do it, and ended up being the largest COVID testing center in the country at lower cost and higher speed than anywhere else in the country, indeed probably the world was doing. And I remember that night when someone had to hit the button and stop the line. It was right before Thanksgiving.
Yes.
The first year, right? So we're doing all the college testing. All the college testing, all the students want to go home for Thanksgiving for the first time. And we had to stop the line because I think everything was coming back positive for a couple hours, which is
Not
not normal.
everything. That
More than normal.
Too many things, yeah.
And we got a lot of calls from angry parents and angry students who couldn't get their tests quickly, but it was resolved really quickly because I think one molecule had infected the
Well, it's
entire
an incredibly overly sensitive test, very sensitive test, and so yes, any little tiny bit of contamination, like just a couple copies of DNA floating around that shouldn't have been there, get amplified because it's PCR, right? So yeah, that kind of test is very hard to scale and to do it well and accurately, like we were able to do. And David, I know you questioned that a lot. You were like, do you really want to do PCR at that scale?
No, it was great.
It's so sensitive.
Right.
Yeah.
It was great.
But no, it was something.
One thing just to connect dots is we're talking now for the last few minutes about scaling activities and execution, all that stuff. But I think also there's a lot of learning for students and postdocs, and there's opportunities in learning for students and postdocs in academic labs. There's, first of all, just the access to the technologies, right, and the ability to envision experiments that you couldn't do. But I know that I had, wonderful lab, and people have gone on to do amazing things. And they always talk about how, being in an environment where like people could dream of things, build them, scale them, just learning how, watching, you know what I'm saying? But not only having access to it, but it influenced how they thought. And if you look at most academic labs, there'd be like a student or postdoc who does something, and then when they leave, it's like you ground to a halt and then you start again. It's actually highly ineffective. So I remember some of my former people saying, just like learning from watching, Stacey and the Broad, that like you should actually have something called they didn't call it an SOP, but you should not just like have an individual do it their own way. You might want to see that people can actually replicate it. So like when you see that, people say a lot of the data in academic labs is irreproducible, that's probably because like one person did it one way and no one can reproduce it. And so we would do things in my academic lab, not the part of it that was doing genomics, of just like, we better have someone else try that experiment. We better have someone else analyze the data, that I think came from being in an environment where that type of quality was being used. So it's not just the access to the technology, it's a way of thinking about how do you generate results that are actually reproducible.
So let me turn to Todd for a second, because we should talk about cancer. Cancer is an amazing Broad story in that we've made a lot of progress both understanding cancer, but also influenced the way people can even learn about their own cancers and even approach treatment. And we have Matthew here in the audience too. Bill Sellers couldn't make it today, but Todd, maybe you kick us off, and maybe Matthew can fill in some gaps after.
Yeah, so I do want to turn to Matthew in a second, but I want to add sort of a perspective for those that are newer to the Broad, to sort of help you understand what was cancer research like before Broad and before the Human Genome Project, which is, there was basically only one way to approach a cancer research project, which was to say, step one, start with a hypothesis. Step two, do an experiment to try to prove that your hypothesis is correct.
That sounds...
whether it is or not, try to keep working to show that it's correct. The idea that you could step back and say, I don't actually have a very good hypothesis. I don't know, I'm not smart enough to know what the right answer is. I want to just collect data comprehensively, step back and ask, what do the data tell me is important about this particular tumor, for example. That was just like a radically different way to approach the problem. We take it for granted now yes, that's how you would approach any kind of interesting biological or clinical question, but that was not how it used to work. And so the first effort that I'm aware of to really systematically say, I don't know what genes are mutated in cancer and are therefore causes of cancer. Let's try to measure them all. Sequence them all. Well, that time it was impossible to sequence them all, so start with a small subset of the genes. And maybe turn to Matthew, because this is also the 20th anniversary of what is arguably one of the most important discoveries in oncology over the past probably 50 years. So Matthew.
Sure. Well, thanks, Todd. So Bill Sellers and I both joined the Dana-Farber faculty in the late 1990s. And we were both already inspired looking at what Todd was doing, because Todd had already the vision, okay, you can actually systematically study cancer. You can look at gene expression arrays, and you can do an unbiased way, and based on that, you can build a new and powerful classification of cancer. And suddenly, out of the genome, comes this new vision of what cancer is. And at the same time, Stacey mentioned coming here and looking at SNP arrays, and Stacey, David, and Eric were working on SNP arrays, and we said, okay, we can now start looking at the somatic genome and say, how is that different from the normal genome? Cancer is this development from our germline genome to a totally different genome, and that genome provides a vulnerability for cancer. So Bill and I were starting to work on SNP arrays and gene expression arrays, and then we were reading about the first sequencing of the human genome reported at the start of 2001, and what were we going to do with this new information? And Bill and I took a plane ride to Europe, and we stayed up all night talking. And we were talking about what subset of the human genome could we look at, and Bill said, let's look at the protein kinases. There's this new drug, Gleevec, it's treating leukemia. Let's look at the kinases. So we started an ultra-high throughput concept, and we couldn't do it by ourselves, so we went to Todd and Stacey and David and Eric and said, look, we want to do this ultra-high throughput experiment. We want to look at more than 100 cancers, and we want to look at almost 100 genes, and we want to look at 2 exons in each of those genes. So we were going to look at 20,000 bases in a couple hundred cancers and normal controls, and this was like unimaginable scale, and we needed some computational help, and so like Stacey and David introduced us to like their crack computationalist, and so this unimaginable scale, this is probably like, a millisecond of sequencing here now. And so then we found our first really major discovery, as Todd alluded to, was mutations in the epidermal growth factor receptor gene in lung cancer. And that's just led to an explosion of targeted therapies in cancer. And what does that mean for patients? So when we started this work, there were 160,000 people in this country alone dying every year from lung cancer. Today, it's 125,000. So that's 35,000 more people living today. And there was a paper from the National Cancer Institute in the New England Journal of Medicine a few years ago saying much of that decline is attributable to the genome-targeted therapies. So a little bit like the Broad Institute, this was just the beginning, right? This was an example. And then Todd and Eric and Bill and others started envisioning, well, let's go from this to actually sequencing all the genes in all of cancer. And then Gadi Getz was in Todd's lab as a postdoc, and Gadi really became a driving force on the computational analysis of the genome. Stacey, the driving force in making it possible to scale up experimentally. And we went from sequencing, just 2 exons out of a small number of genes to sequencing all the exomes in the genome and then the whole genome in 10,000 samples. And I actually developed a new rule to understand what type of gene is responsible, their mutations in cancer. It's a really simple rule. If I thought that class of genes wasn't important, it was really important in cancer. So like chromatin regulators, I thought, there's no way that's important. They're mutated in cancer. Splicing factors, they're mutated in cancer. Immune regulators, they're mutated in cancer. And this has led to a continuous explosion of therapies. One of the other parts of the genius of the Broad was that unlike all the other genome sequencing institutes, right? So, the Broad brought together genomics and the hospitals, and it also brought together chemistry and drug development. And, I think Stuart Schreiber pioneered that, but we'll probably be hearing more about CRISPR today and, the Broad's role in CRISPR and the Center for the Development of Therapeutics. And as part of all of this, we've got a collaboration with Bayer that's been running through CDOT for the last 11 years. And so I'll say something that most people in this room don't know about because it just happened on Monday, which is we've developed an EGFR and ERBB2 inhibitor together with Bayer, and clinical trial results were announced at the American Society of Clinical Oncology. On Monday, this is work that's been led in large part by a professional scientist, Heidi Grulich, who's been in the cancer program for 20 years. And this drug developed jointly between the Broad and Bayer has a 70% response rate in a group of patients for whom there was no treatment before. So we see this arc from genomic discovery to drug discovery to benefit for patients that happens both within our walls and beyond. And I think this is, again, just a really unique feature of the Broad is the expansiveness, and that goal to bring technology to use for the benefit of people.
So there's a third video. This is the second to last one, I swear. And this is, we spoke to a lot of Brodies, not just the front row people, but what they think the impact of this community has been and will be over the next, the next couple of decades. And so let's show this next one, and then we'll react to that after that.
My own experience was the thrill and the excitement of doing something new and big and different with people who also wanted to do something new and big and different.
It really was a great time to be part of this nucleating center, which really has just grown ever since then and spread to other fields beyond genomics.
Genomics was the only game in town in terms of trying to drive things forward, and now we have metabolomics, proteomics, and maybe much more importantly, the integration of all that information.
I think the things that we almost take for granted today, they were almost unimaginable 20 years ago or so. I mean, who would have imagined a world in which at our fingertips we would literally have more than 1 million genomes from humans that we can just start analyzing?
We are still at an early stage, taking these basic discoveries and understanding what it will take to turn those into biomarkers and treatment.
Not only generating the data, but fully interpreting that data for patients to make diagnoses, to inform treatments, and that's been an evolution at the Broad.
I think the Broad was really one of the founders of the Human Cancer Genome Atlas Project, which eventually resulted in the sequencing of over 10,000 human cancers.
I can't think of a cancer project today that doesn't somehow refer to TCGA data and saying, oh, let's check this result in TCGA, or let's use the TCGA data.
Without the Broad and without the Genome Project, that simply never would have happened.
COVID-19, 1 in 20 diagnostic tests and 1 in 20 genomes in the United States were run at the Broad. One institute, one organization, one lab doing a significant fraction of the country's sequencing is pretty amazing.
Broad has allowed us to gain the critical initial molecular clues to psychiatric disorders that have now sent us on our way.
Genomics has given us a lot of clues. The hard work continues to figure out what those clues means. And then from there, we hopefully can make an impact onto how to fix those problems.
The time is ripe if you're systematic about sequencing genomes, about figuring out gene function, about finding chemical and genetic modulators of those gene functions. It's only a matter of time before that really starts to flood the clinic, I hope, with new treatments.
That ability to say we're gonna get to the mountaintop, but we have a lot of work to do to get there is why the Broad is the Broad.
If none of the challenges seemed difficult, we wouldn't have anything to do. I mean, that's our stock in trade, is coming up with totally new ways to do things that used to seem impossible.
What has to stay the same is this excellent mixture of different kinds of people who complement each other. There's nothing here that's done as a solo act, and I think that's very important to convey to new people coming in.
There's just so much more that we have to do, to discover, that that excitement has not left me in the last 20 years.
When you find a home, scientific home, and it continues to deliver new and fascinating things to work on, and continues to focus on tackling the most important problems, why would you go anywhere else?
Steve Carr for the win there with the last quote. So going back to where we started, 1999, you're standing there in the Atlantic Room giving a speech saying, this is history. A lot of history has happened since 1999, just in this community alone. How do you reflect back on that, and how do you think about where we're headed from this point forward?
So why was there a lot of history made? I think the Broad has always worked hard to have a clarity of vision. That's actually really hard to do because your initial vision is murky. And what I love about this place is folks are so social and so stubborn that they can go back around and around and around, arguing about where is the world going to the point where you distill it to something that's really clear. And that can't happen without being part of a community. I was originally a pure mathematician, and I left pure mathematics because it was such a solitary, such a monastic kind of career. You did most of it in your own head. And I eventually concluded I want to be part of something bigger than myself. That's the most, and I think it is one of the most satisfying things in life, is to be part of something bigger than yourself. I think we've got a community here which every several years is prepared to go back and ask, where does the future need to go? Not where does my grant have to go, where does the Broad have to go, where does the world have to go? And we're willing to talk in that way. And we've seen waves of this, from the early stuff on the back porch to things involving lots more people and lots more ambitious things, and the whole creation of single-cell biology, in my opinion, single-cell genomics arising here, and so much to do with CRISPR arising here. You ask, where's the future? The future is the same. I think we're just ripe for that again. It goes in waves, and I think we're at the point where there are a lot of these amazing pieces lying around. We're seeing these therapeutic impacts, and we're seeing ways to think about the programs of the cells and all that. I kind of feel like the next year or two, the Broad is again going to gestate and give rise to these kind of clear, exciting visions, and not be daunted by, where do I get a grant to do this? Or, oh my God, it might be hard to do it. They're going to say, what is the thing that needs to get done? And so I'm not going to prognosticate what it will be. I have opinions, but I think it's that magic ability to do it and to say, if it requires big scale, well, we have the model of platforms. If it requires lots of perspectives, we have the model of programs. They all interact. If somebody isn't here, well, we have a model where anybody from the entire Boston community can be drawn into this. We have all the tools at our disposal to think big, and therefore we have no excuse, because there aren't that many places where you could do it, and so it's on our shoulders to keep inventing that future.
You know, another answer to that, in addition, and you might remember in 2008, there was something at the Harvard Business School looking at different institutions, and Eric was asked, or whatever, we were there, and said, what do you think the most important, I've struggled to remember the word product, what will be the most important thing? Remember this? The Broad does. And Eric said, without an instant, he said, the people we train. And I think that was such a great answer at the time, and it's so true, that we haven't talked about it, so I just think, and it's because you're some of them, those people, right? Is that there have been thousands of people who've gone through here who have had impact here, that's wonderful, that have had impact in lots of other places, that have gone on and trained their own people, whether it's in academia or industry or government. And so when we think about the Broad, in addition to whatever it is that we do here, I guess I'll say we, I'm still part of the Broad.
Always the Broadie.
But I hope so. But in addition, there's what all the people here learn, think about, decide to do. And I don't think that should be underestimated. I remember, everywhere I go, people want to ask me, how do you make another Broad? And it's pretty funny, because one of the people who lives in New York City, when we were announced, hated the Broad, and actually wrote, it was in a newspaper saying what a bad idea the Broad was, and then 10 years later tried to recruit me to run the replicate of the Broad. And but if you go around, every city wants to understand, how do you build a Broad? And the reason is because, one, it does stuff, but also because it's a wonderful place to train. It's a wonderful place to have things grow out of it, whether it's academic labs or companies. And so I think that, again, I just sort of, I agree with you, but when those ideas that Eric's talking about, some of you might have them, and you should say, how do I figure out, one, what we do, but also gather a team, as you said, beg forgiveness, don't ask permission, and then you'll gather a team, and then you'll train the next generation. It's it's even more than what's in this room. It's where those thousands of people have gone, and what they've done.
Right, and they become empowered by knowing that they can do it.
No, they take it with them.
Yeah.
The sort of the idea that I can do something I didn't think I could do. I could gather with other people. I might be able to scale it. I might be able to change it. Maybe we did it one way and they want to do it a different way. That is a lesson that you never, Francis said, once you've been landerized, it never goes away. You can take it and do with it what you want. But I think it's that willingness to believe and that willingness to act and to build a team and to go achieve.
Yeah, and in the beginning it was getting landerized. That was actually the term for it. But over time, it's infectious, and everybody does it to each other. And that's the true power, is that this has just a tremendous spread, and Brodies know how to train and unleash the imaginations of other Brodies, and that's why it's worked.
I actually want to ask Stacey a question about that because all of us have day jobs, right? We all get drawn into stuff every single day, whether it's email or work at the lab or whatever it is. What advice do you have for those of us here in the crowd to continue to think big and try to push to build big collaborations and figure out where the field needs to go while at the same time getting dragged into the daily stuff we all do?
Lee, that's a hard question.
Yeah. Sorry.
Well, first of all, I think great teams is like my very first answer because there's so many talented people we work with who are they're keeping the trains running all the time while we save that little bit of time to think about what's next. And what I really like to think about is how we're building stuff here for the Brodies want, but like part of that building is always, but someone else is going to want it too, right? And so doing it in such a way that like we can make it available to the world, or we're using it to build some big data set, some data resource that other people are going to tap into, and then we're going to use that over and over and over again for the next set of experiments, if that makes sense. I don't know. But that's sort of the way I think about like how we decide what we're going to spend our energy in building up here, like what we might want to use again and again and again. Yeah.
Anybody in our front couple rows or anywhere want to talk about that? I'm staring at John Dench.
Oh, yeah.
And maybe Pardis.
Sure. So I guess my origin story at the Broad is I was not like in the band, but I guess I was like an early groupie. I don't know. I was a graduate student at MIT as the Broad was getting started in a very traditional lab. And as David kind of mentioned, the first time I ever really heard of the Broad was, hey, I've heard there's this place that has really good food. They have like salmon for breakfast.
That was intentional.
And so I was like, well, where is this? Place 320 Charles. Well, how do you get there? I don't know. We should go find it on MapQuest because Google Maps didn't exist yet. So, we figured out how to get to these program meetings, and there was good food. And at the time, I was trying to figure out how RNAi worked, and I'd heard of this Human Genome Project. I had TA'd for Eric. My wife TA'd for Eric. You get no credit for our relationship, but we shared that in common. And then I was like, oh, well, what if we could use RNAi at scale to understand what all these genes are doing? And there was a lot of work thinking exactly as Stacey was saying, how do we scale this? How do we do this as a team? That led to the RNAi platform. It led to now a lifelong collaboration with Dave Root and many here at the Broad. But the idea that you can build things here and maybe early on in my career when I would see people using tools and technologies and approaches that I had built, I'm like, hey, my name's not on that, I would get ticked off. Now I've realized like that is the coolest thing you can get to in science, when you're making contributions and people don't even know that you exist on some level. I think you've really made it. And I think that the Broad, we continue to do stuff that we put out into the world that has impact that we never get to see as Brodies, but it is something that is happening the world over.
Thanks, John. And so Pardis and I have talked a bit about, she thinks big and builds collaborations. And I know, Pardis, you've mentioned a while ago that you found a home in the Broad that thinks the same way sometimes.
Yeah, I mean, so I'll start with a little bit of my origin story also, which is, well, the true origin story is I stalked Eric and I made him my undergraduate advisor as an MIT freshman. So we are at 30 years now where he's been my advisor, which is wild. And I'd still say you're my advisor. I literally will always look to you for advice. But the one thing I'll also say is it's really fun for me to see this group of people here because I remember the first time when I came back to work in the lab, David was my first meeting. And David, like again and again, would come into my life. Like David, I don't know if you know, I quote you all the time. Like there's so many moments in my life where I'm like, I just quote something you said that how it impacted me. So like this place is amazing. It is a family, and it is, and it does hard problems, and it gets in, fights and how to do it right. Like one of the things that's interesting about like what we're sort of talking about is we do go really, really fast, but we're also like infuriatingly slow sometimes because we want to get it exactly right. And I think that, Niall kind of explained circumstances like that, but it's that we move as fast as we can without breaking things. Like we don't want to truly break things. We want to figure out how to move. And we're doing that right now with H5N1, and Lee and I are in something. So fundamentally, that is what is remarkable about this place, is it takes on challenges. It says anything is possible, but it says it really requires incredible hard work and thoughtfulness to it. And for us, I cut my teeth in human genomics as Eric's mentee, but then also as a postdoc with him and with David and with the whole crew. Actually, it is like team mentorship here too. But then when I moved into infectious disease, I often say I'm just coasting off of what we do in human genetics, right? We're in this space where we can take all the tools and all the advances, and we just kind of go around like a kid in a candy store and say, we could apply this to infectious disease and that to infectious disease, and not just the technologies, but also the mindset. Like one of the things we did during the Ebola outbreak was just release our data as soon as it came off of the sequencers, and that was shocking. I can't tell you the number of people that like reached out and were like, this is a mistake, right? Like, where's the paper? I don't understand what's going on here. But we infuse that without even knowing it into every part we do. And I'll say the last thing is where we're at right now, I often talk about infectious diseases. We are at a place where the Human Genome Project, where human genetics was before the Human Genome Project, where you're doing things on a single gene basis and you're trying to, figure out what's going on. And now we're doing things on a single infectious basis. It's COVID or not COVID. I mean, how can we ever develop therapies if we don't know what most people have when they're sick? And so that same mindset where I see the Broad going is transforming field after field by infusing that scale and collaboration to make it happen.
So it's a great story, but just to give the perspective from the other side, Pardis and your lab are examples where data was collected and a continued identification of a problem that could be solved. So I almost see it the other way. You're saying you're a kid in a candy store and you're coasting. I remember when you first came back, it was like, wow, you might be able to study natural selection from this population genetic data. You published this beautiful paper on that, and then you started continuing to study that and said, oh, it looks like natural selection might involve infectious diseases. And then you started moving into infectious disease, but then you were still doing it in the lab, but then you started going to places and actually being there as part of a crisis and bringing the technology to them. So I would almost turn it around, and again, my message to everyone is like, what you should do is like, some people will build this scale and some people will do it, and some people will see the data and they'll see an opportunity, and you also you have the best Christmas cards, but you've trained so many people. I can't tell you how many places I've gone and someone like comes to Vertex for a summer and how did you get into science? Oh, I took a course from Pardis. So there is this virtuous cycle, but there's also, for those of you out there who don't run a platform, right? Look at the data, think about a problem, figure out how you might move it forward. That's the virtuous cycle. So you could be either one of these. That's why you want to be together, because you can do that.
Yeah, can I add to that? I mean, so much of what we talked about today and a big part of the Broad is highly technical and it's about scale. But what I think is in many ways most galvanizing for the community is to step back and say, like, what do you really want to accomplish? What are you trying to achieve? So like following on what John Dench was talking about, you can think about RNAi and CRISPR functional genomics from a highly technical perspective, or you can say, actually what I really want to know are, what are all the vulnerabilities? What are all the Achilles heels of cancer, of all cancer types? That's like a very non-technical statement, but it's really motivating to say, we could actually know that. And then that becomes the end, and the rest is a means to get there. Exactly. Where you got to work out a lot of stuff, you've got to figure out how to make it cheaper and high quality data and solve a lot of technical problems. But having that sort of big idea that is the ultimate goal is really motivating for everybody.
And just another like favorite story. Eli and Edie were visiting. Must have been I don't even know where we were the Broad yet, but it must have been. But like they were visiting, we were in 320 Charles, and we were going to bring in some young people, postdocs, to tell a story. And one of them was going to talk about, I think it was with Stewart at the time, and he was talking about saying chemical biology, but he happened to be a transplant hematologist. And the other one was Steve McCarroll, who was my postdoc at the time, and he was working on structural variants in the human genome. So we chatted with Eli, and then we said, we're going to bring in these two postdocs to tell a story. And they walked in, they said, I hope you don't mind, but we were sitting out there waiting to come in, and we came up with a much better idea than either of us had planned. So do you mind if we tell you our idea? And we go, I guess, like talk about begging forgiveness. We're in front of our donors, and they go, well, we were talking about how Steve was studying how certain genes are deleted from the human genome, and it turned out Jay Bradner, who was the other person who later went on to become now he's head of R&D at Amgen and Novartis. He was saying, well, if you delete a gene and you do a bone marrow transplant, maybe that would be a transplant antigen. It would lead to graft versus host disease. They had designed the entire experiment
while waiting
outside the room, and they said, and if we could just get our hands on like 1,000 samples and we could genotype all these things, we could see if that's true. And you can look up the Nature Genetics paper, because I think in the room actually the idea was like, go do it. And then it turned out we had some Finland collaborators. Do you remember? So to get the samples, we worked with the Farber, but also worked with Finland. It turned out the hypothesis was true, and it led to a Nature Genetics paper. And I always loved that story, or the clonal hematopoiesis story. I'm sure there's many others, because it was like two postdocs sitting there. It was like, tastes great, whatever, chocolate and peanut butter, if you know what that is. Like they had two very different fields. Someone working in Stewart's lab, who was a transplant hematologist. Someone who's a human geneticist studying deletions, invent a project that no one else would have done.
So one last thing just to share on the screen, and then we can reflect from that. We asked a couple of the Brodies who have moved on. You mentioned we have alumni out in the world. One of the most important products is people. We asked a couple of them what they found special about the Broad that we want to make sure we protect so that the next couple decades are as productive, if not more so, than the first.
What's special about the Broad is that it is at the same time as boundless as the universe and as small as a village. You can imagine anything here and do anything, especially things that have never existed anywhere. But at the same time, you do it by using this power of a village, of a small community where everyone knows everyone knows what everyone else is doing, and everyone comes up with clever ideas on behalf of everyone else in solving these difficult problems together.
I think people come here, they're inspired to do their best. Inspired to pick really important problems to solve, and given the freedom to solve those problems. That's really what's going to be important as the Broad moves from the first 20 years to the next 20 years. And I'm very confident that this culture of mentorship, inspiration, nurturing is going to continue.
So in the 3 minutes we have left, real quick, what are you most proud of so far?
Moi?
Go for it.
Well, I think it is this culture of it. It sort of transcends technology, diseases, specific questions. But if you've got a group of people that really want to work together to just solve a problem, not write a paper, but solve a problem, you can do anything. And if that persists into the future for the Broad, as I'm confident it will, the rest will follow.
You're not supposed to say what you're most proud of, because that's like picking your favorite child. Eric taught me that. I can say what I'm particularly proud of, I suppose. Right? See, I learned. I think I'm particularly proud of, I think, the resources that we've created for the world, right? Like creating things like the Cancer Genome Atlas, or the HapMap, or the Human Genome, whichever, pick any of them. Because, I think we did it, for the greater good. We did it for the community, and we learned a lot from it. And we didn't need to, all have our name on every paper, but it's out there for everybody to use. I think, I think that's I'm particularly proud of that.
That's a tough one. I mean, I guess what I'd like to say is it's the impact on medicine, because there are so many things, we did talk about and didn't, at Vertex, we've made medicines that came from genome-wide association studies and CRISPR, and there's hundreds of examples all over of that. But I was thinking, what's the honest answer? And the honest answer is like the culture and the people. And like when I think about it, when I drive by, or I walk by Kendall Square and I, see students or people, I meet people all the time and they're like, oh, I worked at the Broad. My favorite line was someone once said, you know, I worked at the Broad and then I went somewhere else in Boston, I won't name. And they said, it was like, you know in the movie The Wizard of Oz when you go from like black and white to color? That's what it was like going from the other place in Boston to the Broad. And hearing all that excitement and all that energy, that's what I'm most proud of because one, it just matters to your heart, but also because of people hopefully, when I was young, I was getting cynical when I met you. That like, I was 33 years old, I was an MD, PhD, 14 years of training. What was I gonna do that mattered? And I told you something, and you said to me that thing, you said, what do you really want to do? And I told you I want to find the genes for complex disease, but everyone tells me I'm an idiot, and it's not possible. And you said, well, what else would make sense? And to be here like 25 years later, there's a song by Bob Dylan that goes, but I was so much older then. I'm younger than that now. And I hope you all feel younger, and you feel the possibilities so that you can go and dream your own dreams and make them happen.
I agree.
I don't think I've ever done that before. He didn't say anything.
No. Exactly. It's the number of people whose ambitions and minds and hearts have been unleashed to take on whatever they think is really important. So much in the world constrains people to think narrowly, what can I do? And the Broad is about, I mean, NIH reviewers sometimes complain, oh, the proposal is too ambitious. That's ludicrous. There are a lot of things you can say about a grant, about why you shouldn't fund this grant, but that it's too ambitious. We should all be trying to be too ambitious. And we have a culture and we have people and we have each other that lift each other up to do the things that matter in the world. And we can do it because we have each other to help us and to learn from. And I think that's a really special thing. If I think about what am I proud of, what are we all proud of, to have a culture and a people that does that and sustains it and has done it for 20 years and is thinking so hard about how do we preserve that for the next 20 years, that's really magical. And like I said in the Atlantic room, I mean, you're all still part of history, and you're all creating that history. And there aren't that many places and things you can do in life where you can do it. And it's really special. So I want to say thank you to everybody here, everybody there, the other 23,000 people who aren't here. What a place, what a mission, and what a responsibility that comes with it.
So in the tradition of Broad Institute, I can already hear the people who are watching from the overflow room starting to line up for food in the line right now. So Todd, maybe you can close us out.
That was a lot of fun. Thank you all for being here. Thank you, Lee, for pulling us all together. I hope everyone feels proud of being part of this community and is excited about what we can do together in the years to come. In that spirit, there's some kind of a device out in the lobby near the bar, I think.
Post-it note.
A Post-it note?
Yes.
Okay. I've seen that before, a large Post-it note, where we encourage all of you to scribble down your ideas about what might the next 20 years of the Broad Institute bring to our community. So bring your ideas. They're all welcome. Hope for those of you that are here in the building, you'll join us for reception out in the lobby. Thanks, everybody.